Okay, so today we're really diving deep into the world of synthetic biology. We're going to be looking at Teselagen's Advanced Molecular Biology Toolkit. You sent over some excerpts from their documentation.
Yeah.
And it sounds like they're really kind of upping the game for
Yeah.
anyone who's working with DNA. So imagine, if you will, having this platform that not only helps you design genetic sequences, but streamlines like the entire research process.
Yeah. That's exactly what they're aiming for. Synthetic biology is all about manipulating this genetic material to kind of solve real world problems.
Mm.
And so this toolkit is designed to make that whole process faster, more efficient, and a lot less prone to error.
Yeah, and one of the things that really jumped out at me when I was reading this was just their emphasis on visualization.
Hmm.
They have this really cool feature called Visualize and Export Genetic Schematics. And it's essentially like creating blueprints, but for DNA.
And having those visuals is really, really crucial in synthetic biology because, we're talking about complex genetic designs with multiple components, and having this standardized visual language ensures that everyone's on the same page. It's really vital for collaboration, for publication, and even just kind of wrapping your own head around these really intricate systems.
Yeah, it reminds me of how architects use blueprints. You can't just describe a skyscraper. You need to actually have those visual plans to see how everything connects. And speaking of connections, let's talk about dynamic rule set creation. This seems like it's designed to eliminate those like heart-stopping moments when you realize a tiny error could snowball into this like major setback.
Right. And in a field where precision is just so important, Teselagen has taken a page from industries like aviation and finance that really rely on these really sophisticated systems to try to minimize human error as much as possible.
Yeah, and in the document, they talk about tagging DNA parts with specific labels like backbone or coding sequence, which is often shortened to CDS.
Exactly. So by establishing these rules within the software, you're essentially creating the safety net. So let's say, for example, you can set a rule that says that every coding sequence, the part of the DNA that actually gets translated into a protein, always starts with the correct start codon, ATG. So it's kind of like having the superpower assistant who is double checking your work in real time, making sure that you don't accidentally swap out a crucial component in your genetic circuit.
It's fascinating how they're using software to almost like hardwire those best practices into the design process itself.
Uh-huh.
And speaking of analyzing designs, the sequence alignment feature seems incredibly powerful. It's like having a detective's toolkit, but for DNA.
Absolutely. It's like comparing fingerprints, but at the molecular level. So sequence alignment allows researchers to compare DNA or RNA sequences to understand how they're related to each other, to identify any differences, and to even trace evolutionary lineages. So this is used in a really wide array of applications, from tracking disease outbreaks to, like you said, optimizing genetic designs.
And they highlighted a specific tool for this called the Dapper tool for parallel part alignments.
And what's really fascinating about this is that it allows for comparison of multiple sequences all at the same time, which really speeds up that process. So instead of comparing two fingerprints at a time, you can analyze an entire database to find the closest match or even pinpoint these very subtle variations.
Hmm.
And this is really valuable in synthetic biology, where researchers might be tweaking and optimizing these genetic sequences to improve their function.
So it's not just about matching, it's about understanding like the nuances of those matches, finding those tiny discrepancies that could hold the key to some sort of breakthrough. And when it comes to dealing with these massive amounts of data, it seems like Tesselation really has that covered as well, especially with advanced NGS reads mapping, although I'll admit that one sounds a bit intimidating.
It does sound very complicated, but it's actually really interesting. So next generation sequencing, or NGS, it generates this just tremendous amount of data. Imagine a puzzle, but with millions of pieces. So this feature helps researchers piece together that puzzle quickly and accurately, making sense of just this vast amount of genetic information.
So instead of being overwhelmed by this sea of data, researchers can actually focus on extracting those meaningful insights. It's about working smarter, not harder.
Precisely.
Yeah.
And speaking of working smarter, there's a quote in the material from Dr. Michael K. Pleik, who's the vice president of synthetic biology at LanzaTech. And LanzaTech is a company on the cutting edge of carbon recycling. They're literally transforming pollution into products.
Wow.
And Dr. Pike calls Teselagen's platform an essential foundation for the work that they're doing.
It's so cool because, we often hear about like the potential of synthetic biology.
Right.
But this feels like a really tangible example of how these tools are moving from these theoretical possibilities to like real world solutions. Like it's not just about creating new things, it's about addressing some of like the biggest challenges that we're facing as a planet.
Exactly. And, while LanzaTech is focused on this carbon recycling, This toolkit has applications across a wide range of fields. So imagine developing personalized medicine that's tailored to an individual's specific genetic makeup,
Wow.
or engineering microbes to create biodegradable plastics, for example.
It sounds like science fiction.
It does, but it's becoming increasingly realistic.
That's amazing.
Yeah, we're entering an era where we can harness the power of biology to create solutions that we once thought were unimaginable.
Yeah, absolutely. It's really cool. And it seems like Teselagen has considered, the needs of researchers who are working at the forefront of this revolution.
Yeah.
So, for example, they have a feature for Sanger sequencing and chromatogram visualization.
Yeah.
And I'll admit that Sanger sequencing, that's something I haven't heard of in a while.
Yeah, so Sanger sequencing is a more traditional method of DNA sequencing, and this tool allows researchers to visualize those results, which can be really helpful for spotting any potential errors in the data.
You see, with these cutting edge technologies, having those reliable, tried and true methods is still really crucial.
Absolutely. Think of it like a carpenter who still relies on a trusty hammer,
Right.
even though they might have access to all the latest power tools.
Right.
Sometimes those classic techniques are just the most effective for certain tasks.
Mm-hmm.
And being able to seamlessly integrate those methods into a platform alongside these more advanced tools like NGS analysis really speaks to just the comprehensive nature of Teselagen's approach.
It's like they created a platform that grows with the researcher. So you can start with the fundamentals and then gradually incorporate those more complex techniques as your work demands it.
Exactly.
And that accessibility seems key, especially for a field that's evolving as rapidly as synthetic biology is.
Yeah, you hit the nail on the head.
Yeah.
One of the things that's really impressive to me about this toolkit is its user-friendliness. So they've managed to make these incredibly complex tools accessible to a much wider range of researchers,
Wow.
regardless of their technical expertise.
That's huge. Because by lowering those barriers to entry, you're essentially democratizing access to these really powerful tools.
Exactly.
And that opens up the field to a wider range of perspectives.
Right.
It could lead to a whole new wave of innovation.
Precisely. So by empowering scientists with the tools that they need, Teselagen is helping to accelerate the pace of discovery and kind of drive us towards this future where the seemingly impossible becomes possible.
Yeah, it's really inspiring to think about the potential impact. And it seems like Teselagen is playing a really key role in shaping that future. It's a future where we're not just manipulating DNA in a lab, but we're using it to address these global challenges.
Yeah.
Advancing healthcare, reimagining the very fabric of our world.
It's a future that's just brimming with these incredible possibilities. And I think, what struck me most about Teselagen's approach is their commitment to creating this toolkit that's as comprehensive as it is powerful. It's not just about designing these genetic sequences in isolation. It's about giving those researchers the tools that they need for literally every stage of this entire process, from that initial visualization and analysis to data management and collaboration.
It's like they've considered the entire ecosystem of synthetic biology research. They're not just handing scientists these high-tech instruments, but they're creating this environment where those instruments can actually be used to their full potential.
Exactly. And, by streamlining that workflow and automating those kind of tedious, time-consuming tasks, they're freeing researchers to really focus on what truly matters, which is the science.
It's not just about making the research faster, but it's about making it more intellectually stimulating.
Absolutely.
And ultimately more rewarding for those scientists themselves.
It's about fostering their creativity and really empowering them with the tools that they need to not only keep pace with this rapidly evolving field, but to actually push its boundaries and redefine what's possible.
Well, it has been absolutely fascinating to take this deep dive into Teselagen's toolkit with you.
Yeah.
And it's clear that they're not just building software, but they're building a vision for the future of synthetic biology.
Yeah.
A future where these incredibly powerful tools are accessible to a much wider range of researchers. And in doing so, they're really driving innovation and changing a better world.
And, if this deep dive has kind of sparked your curiosity even a little bit, I definitely encourage you to kind of explore further. Teselagen's website is a fantastic resource, and there's countless online forums and articles and even local events where you can really learn more about this fascinating field of synthetic biology.
Yeah, it's a field that's constantly evolving with new discoveries being made all the time. Who knows? Maybe this deep dive has sparked an idea for a future breakthrough.
The beauty of knowledge is that you never know how it's going to inspire. And if our conversation today encourages even one person to kind of delve a little bit deeper into this world of synthetic biology, then we've really accomplished something remarkable.
Beautifully said. And on that note, it's time for us to wrap up this deep dive into Teselagen's Advanced Molecular Biology Toolkit. A huge thank you to you for lending your expertise to this exploration.
It was my pleasure. Until next time.
And to our listeners, thank you for joining us on this journey of discovery.
Yeah.
Remember, the world is full of these fascinating topics just waiting to be explored, so keep those minds curious. Keep asking questions, and we'll see you again soon for another deep dive.